everyone. I welcome you all for this course of plant physiology and metabolism, where today we will learn about biological nitrogen fixation. So, coming to the introduction of nitrogen metabolism, as you all know that nitrogen has comprises of about 78.084% of total Earth atmosphere. It is essential for plant growth and development. However, in soil, the nitrogen primarily exists in the stable molecular form, that is N2, requiring nitrogen fixation processes to make it accessible to plants. Nitrogen fertilizers have played a crucial role in addressing the challenge of feeding the growing global population, with the Green Revolution witnessing a significant increase in their use, leading to enhanced agricultural productivity. Nitrogen fixation is a vital process that transforms atmospheric nitrogen into compounds like ammonia or nitrogen salts, which are essential nutrients for living organisms. Biological nitrogen fixation is particularly significant as it supports sustainable agricultural practices and enhances soil fertility. Researchers focus on studying microorganisms and plants capable of fixing nitrogen, as their findings contributes greatly to the development of biofertilizers. These biofertilizers offer a natural and environmentally friendly alternative to synthetic fertilizers, promoting healthier soil and crop growth. It's essential to emphasize research and development in biological nitrogen fixation to cater to the needs of farmers, especially those in developing countries where access to conventional fertilizers may be limited. By providing BNF research, we can support agricultural sustainability and improve food security worldwide. As you can see in the figure, that the acquisition of nitrogen from the atmosphere requires the breaking of an exceptionally stable triple covalent bond between two nitrogen atoms to produce ammonia or nitrate. So these reactions, known as basically the nitrogen fixation, occur through both industrial and natural process. Nitrogen stands as a crucial element that can enhance plant growth and production as well as yield. It holds significant roles in various biological processes as a primary component of chlorophyll, that is vital for photosynthesis, and as a fundamental building block of amino acid, that is essential for protein synthesis, then nitrogen is a key constituent of essential biomolecules like ATP, that is adenosine triphosphate, and nucleic acids. It plays diverse role in metabolic functions, facilitating growth, production, and genetic inheritance. Furthermore, nitrogen contributes to the formation of essential compounds such as chlorophyll, cytochrome, alkaloids, and numerous vitamins. Its presence is integral to the sustaining the overall health and functionality of the plant system. Nitrogen fixation, that is the process of converting atmospheric nitrogen into form usable by living organisms drawn from various sources. Atmospheric nitrogen constitutes approximately 78.084% of the atmosphere that serve as a vast reservoir. However, plants cannot directly assimilate this form. Certainly, there is some bacteria, blue-green algae, and leguminous plant that possess the ability to fix atmospheric nitrogen, making it accessible. Alternatively, nitrogen can be found in compounds like nitrates, nitrites, and ammonia, with nitrate being the predominant form. These compounds are utilized by many soil organisms and can also be absorbed by higher plants. Additionally, organic nitrogenous compounds found in insects serve as another source of nitrogen. Insectivorous plants are capable of utilizing nitrogen from these compounds to support their growth and development. Overall, the nitrogen fixation relies on a range of sources, each contributing to the availability of this essential nutrient in ecosystem. As you can see here that there are two types of nitrogen fixation. First one is called as a non-biological or natural fixation, and another one is called as the biological nitrogen fixation. Lightning contributes to approximately 8% of the natural nitrogen fixation process by converting water vapor and oxygen into reactive hydroxyl free radical, free hydrogen atoms, and free oxygen atoms. These reactive species then interact with molecular nitrogen to form nitric oxide, which subsequently falls to the earth with rain. Another 2% of the nitrogen fixation arises from photochemical reactions involving gaseous nitric oxide and ozone, leading to the production of nitric oxide. However, the majority, that is about 90% of nitrogen fixation, is driven by biological process. In biological nitrogen fixation, bacteria or cyanobacteria, that is blue-green algae, convert nitrogen into ammonia, contributing significantly to the nitrogen cycle and ecosystem productivity. As you know that biological nitrogen fixation plays a central role in the nitrogen biogeochemical cycle. This process serves as the crucial entry point for molecular nitrogen into various ecosystems. It involves the fixation of atmospheric nitrogen into nitrogenous salts facilitated by microorganisms. There are two main types of biological nitrogen fixation, that is non-symbiotic and symbiotic nitrogen fixation. Non-symbiotic occurs independently of specific host organism and involves free-living nitrogen-fixing bacteria in the soil or aquatic environment. Whereas symbiotic fixation, on the other hand, relies on mutually beneficial relationship between nitrogen fixing bacteria such as rhizobia and host plant, particularly legumes like peas and beans. Both forms of biological nitrogen fixation are vital for maintaining nitrogen balance in ecosystems and supporting plant growth and productivity. Biological nitrogen fixation or BNF that converts atmospheric nitrogen into ammonia, accounting for about 90% of the total nitrogen fixation, primarily involve living organisms associated with plant roots, particularly prokaryotes. This process is significant as it occurs at ambient temperatures and atmospheric pressure, facilitated by a diverse range of bacteria, cyanobacteria, and symbiotic relationship. Biological nitrogen fixation's role is crucial for providing accessible nitrogen for plant growth and development. Moreover, most of these nitrogen fixing prokaryotes live in the soil, generally independent of other organisms, also called non-symbiotic nitrogen fixation. On the other hand, several forms of symbiotic association with higher plants in which the prokaryote directly provides the host plant with fixed nitrogen in exchange for other nutrients and carbohydrates. Such symbiosis occur in nodules and form on the nodules of the plant and contain the nitrogen fixing bacteria. Now let's discuss about the non-symbiotic nitrogen fixation. So non-symbiotic nitrogen fixing organisms, which are alternatively termed free living nitrogen fixers and can thrive in both terrestrial and aquatic environment. These organisms carry out nitrogen fixation independently and are classified into three main groups, that are the aerobic, anaerobic, and blue-green algae. Among the free-living aerobic nitrogen fixers are azotobacter and Bergeracia. Examples of free-living anaerobic bacteria fixers include the Clostridium and Rhodospirillum. The blue-green algae, also known as cyanobacteria, encompass various types such as unicellular species like Syncoccus filamentous, species without heterocytes like Oscillatoria, and filamentous species with heterocysts like nostoc and anabina. So this slide describes about the symbiotic nitrogen fixation. So basically, the symbiotic nitrogen fixation refers to the process of microorganisms in the soil fixing free nitrogen while living symbiotically within plants. The term symbiosis was introduced by DeBerry and encompasses three main categories: nodule formation in leguminous plants, nodule formation in non-leguminous plant, and non-nodulation. In nodule formation in leguminous plant, certain microorganisms, typically rhizobia bacteria, establish a symbiotic relationship with leguminous plants such as peas, beans, and clovers. These bacteria reside within nodules that form on the roots of the plant. Inside these nodules, the bacteria fix atmospheric nitrogen, which is then utilized by the host plant for its growth and development. However, some non-leguminous plants such as alders and certain tropical trees also form nodules on their roots containing nitrogen fixing bacteria. These bacteria contribute to nitrogen fixation within the nodules, providing the host plant with a source of fixed nitrogen. In non-nodulation category, certain plants do not form nodules on their roots but still engage in symbiotic nitrogen fixation. Instead of nodules, the microorganism may colonize the root surface or inhabit the intercellular spaces of the root tissues to fix nitrogen symbiotically. Overall, symbiotic nitrogen fixation plays a crucial role in providing plants with a vital nutrient source and contributes significantly of soil fertility and crop productivity. Symbiotic nitrogen fixing systems are prevalent in wide range of plants encompassing approximately 200 leguminous and over 200 non-leguminous vascular plant species. Additionally, symbiotic relationships extend in organisms such as azolla, lichens, certain fungi, and grasses. In leguminous plants, nodules typically form on the roots, hosting various species of rhizobium bacteria as symbionts. For instance, in Sesbania rostrata, nodules are situated on the stem, and the nitrogen fixing organism present within these nodules is Azorhizobium colinodans. In non-leguminous plants like Alnus, Cadurina, Myrica, Portia, and Sarcocarpus. Nodules develop on the roots wherein members of actinomycetes bacteria resides responsible for nitrogen fixation. These diverse symbiotic relationships highlight the versatility and importance of nitrogen fixation in facilitating plant growth and ecosystem functioning across various plant species. The next one is Psychotria species, which is known to form nodules on its leaves, hosting the nitrogen-fixing microorganism Klebsiella. Cycads, on the other hand, develop nodules on their roots, with several species of blue-green algae responsible for nitrogen fixation within these nodules. Grasses employ a unique strategy where they produce a mucilaginous sheath on their root surface. Under this sheath, nitrogen-fixing bacteria belonging to the genus Azospirillum reside. So this mutualistic relationship between Azospirillum bacteria and grass roots is commonly referred to as the Azospirillum grass relationship. The nitrogen-fixing microorganisms residing in nodules have the capacity to fix nitrogen at a significantly higher rate that might be around 100 to 200 times more compared to free living microorganisms. This enhanced efficiency stems from their ability to sustain nitrogen fixation over extended periods, typically lasting 30 to 40 days. During this process, nitrogen is converted into asparagine. In the case of azolla, nitrogen fixation results in the production of ammonia. Lichens also participate in nitrogen fixation, with the fixed nitrogen possibly being leached down to the forest floor by rain or becoming available to other plants after decomposition. Legumes play a pivotal role as the primary nitrogen fixing plants in agriculture, highlighting the crucial importance of nitrogen fixation in these plants. This table basically emphasizes the diversity of symbiotic relationships involved in nitrogen fixation across various plant species. It highlights how different plants engage with a specific nitrogen fixing organism and where the nitrogen fixation occurs within each plant structure. This overview underscores the intricate interactions between plants and microorganisms in facilitating nitrogen fixation, a crucial process for ecosystem nutrient cycling as well as in the plant growth. That certain legumes host several species of rhizobium. Nodules are located under the roots. Then sesbania, here the microorganism is the poly azorhizobium and nodules located on stem. So one such as the lichens, the blue-green algae, and the lichens grow on the surface of the trunk and branches of the tree. Then there is a certain fungi act as a host, and microorganism here is the nostoc, and the alga is found in the fungus. So as you can see that in the figure, which shows the filamentous cyanobacterium Anabina heterocysts are specialized cells that play a crucial role in the nitrogen fixation. These heterocysts are found within the filament alongside the vegetative cells. They possess thick cells walls that create an anaerobic inner environment, allowing the cyanobacteria to carry out nitrogen fixation even in aerobic conditions. This adaptation is crucial because nitrogen fixation is an energy intensive process that requires an oxygen free environment to prevent the inactivation of nitrogenase enzyme. By creating anaerobic conditions within the heterocyst, Anabina can efficiently fix atmospheric nitrogen into a form usable by plants and other organisms. This symbiotic association between Anabina and the water fund Azolla highlights the remarkable adaptation of cyanobacteria to perform essential ecological functions in diverse environments. This illustration depicts the process of root nodule formation in three different plant species that are Peptadenia verdiflora inoculated with Burkholderia pimentum. Then we have the acacia species. Here it is inoculated with Cupria vidus, and the next one is Phaseolus vulgaris. It is basically associated or infected with rhizobium. This figure illustrates the presence of symbiotic nitrogen fixing prokaryote within nodules, that is specialized organ formed by the plant host to encapsulate the nitrogen fixing bacteria. The nodules depicted as spherical structure are formed as a result of infection by rhizobium species. These nodules serve as the symbiotic site where the rhizobium bacteria reside and carry out nitrogen fixation in partnership with the plant host. Through this maternalistic relationship, the plant gains access to fixed nitrogen while the bacteria receive nutrients and a suitable environment for nitrogen fixation. This intricate symbiosis between the plant and rhizobium bacteria plays a crucial role in enhancing soil fertility and promoting plant growth. So, the today's session is closed here. In the next session, we will discuss about NOD factor, NOD gene, nodule development, nitrogenase enzyme, etc. related to that of the biological nitrogen fixation.